Thank you. Hopefully you can all hear me. I'd like to just start by thanking the organizers, not only for inviting me, but for putting together this really innovative program that combines facility users, method users, and also method developers. And today I want to tell you about our work using Cryo-EM to build models of axonemes. Axonemes are, of course, the molecular machine that drive the motility of cilia. Cilia are these cell protrusions from eukaryotic cells that can beat rhythmically and are responsible for the motility of many eukaryotic pathogens, including Leishmania, Trypanosome, Plasmodium. In humans, they're also responsible for the motility of sperm. And they can also be found lining epithelial surfaces where they beat not to move cells, but to move fluids over cells. And really the classic example of this is the cilia lining the respiratory tract, which moves mucus in order to remove pollutants, particles like those we heard about yesterday from Victoria, and respiratory viruses from the respiratory tract. Unsurprisingly, because of these kind of fundamental functions in humans, dysfunction of cilia are associated with a number of genetic diseases known as ciliopathies. And these can affect male fertility, also lead to recurrent respiratory diseases and laterality abnormalities where the body organs can adopt mirrored positions. The axoneme adopts or kind of fulfills almost the entire volume of cilia, spanning from the tip to the base, which can be many microns in length. Not only is the axoneme one of the largest molecular machines found in nature, but it's also undeniably one of the most beautiful. And this beauty is particularly apparent in cross-section, thin section transmission electron microscopy. And if we zoom in on one of these cross-sections, we can see that a classical axoneme consists of an arrangement of 9 doublet microtubules that surround a pair of singlet microtubules. Attached to each of the doublet microtubules are axonemal dyneins. These are ATP-powered molecular motors that generate the ciliary motility. And they come in 2 flavors, outer dynein arms shown in red and inner dynein arms in yellow. Bound amongst these axonemal dyneins are a bunch of mechanoregulatory complexes, including the very distinctive radial spoke complex in blue. If we look at a longitudinal section through the axoneme, we can see that it consists of a longitudinal periodicity. And it's not surprising to find that the axoneme, like many large structures, are built of periodically repeating units. These repeat units are quite large in the axoneme. They're 96 nanometers in length. And in 2006, we got the first glimpse of the 3-dimensional structure of an axoneme from Daniela Nicastro's pioneering tomography work. And research in cilia is slightly unusual in that tomography has really led the field, and single particle cryo-EM is now only catching up, which is kind of the opposite of most fields. So this pioneering tomography work and work of others have really defined the overall architecture of this modular repeat of the axoneme, revealing the arrangement of the axonemal dyneins and also these mechanoregulatory complexes, such as these T-shaped radial spokes. But when I started my lab 5 years ago, we really wanted to see if we could obtain higher resolution information that would allow us to build an atomic model of an axoneme. To do this, we needed a model organism. And for that, we used Chlamydomonas reinhardti, which is this green alga. It has 2 beautifully long cilia. And inside each cilia, it has a classical axoneme consisting of an arrangement of 9 doublet microtubules around this central pair of singlet microtubules, with each doublet microtubule split into this 96 nanometer modular repeat bound by dynein motors and regulatory complexes. And Chlamydomonas really is the model organism in the cilia field. And we'll hear more about how Chlamydomonas has been used in cilia structural biology tomorrow from Sam Lacey's gorgeous work. And one of the reasons it's so popular is the cilia can be easily removed, collected, and then imaged by cryo-EM or cryotomography. Now, to get a high resolution structure, we realized we needed to take an alternative approach to tomography. And so we decided to break the axoneme apart into more imageable complexes. And a lot of this work was done with my close collaborator, Ray Zhang at Washington University in St. Louis. So we decided to break the axoneme apart into kind of subcomplexes. And one of the first complexes we focused on was the doublet microtubules, which is really a defining feature of the axoneme. So we took our purified axonemes, we added ATP to stimulate the activity of the dyneins. We added a protease just to break some of the kind of protein links. And here's an image or a couple of images of the microtubules under cryo-EM conditions. So you can see they form very nice microtubules. Some of these microtubules were bound by the doublet microtubules. And using Relion, we were able to attain a 3.5 angstrom resolution structure of the doublet microtubule. And we found that inside the doublet microtubule were a lot of additional densities. These correspond to microtubule inner proteins, which is really an emerging class of protein. We were very fortunate that we had sufficient resolution that we were able to identify these proteins based on side chain densities. And we had to really build pretty much the entire model amino acid by amino acid. One of the great things about Chlamydomonas is there's a pre-existing library of mutants. So we're able to validate our structure by obtaining structures of mutants. This also, as well as providing validation, helped to reveal which MIPS are dependent on other MIPS for incorporation into the doublet microtubule structure. And we care a lot about these microtubule inner proteins because we think they have multiple functions. For example, some clearly provide architectural support, generating this very unique arrangement of microtubules. Others modify the mechanical properties of the axoneme. Others function to prevent damage during ciliary motility and the propagation of the damage. Others provide a uniform distribution of enzymatic activity. And others connect internal and external factors. And I'll show you later that this is important for ciliary motility. However, when we looked at the outside of the doublet microtubule, we were missing a lot of the factors that we know bind there from tomography. However, one factor that we did have were the radial spokes. And using focused classification and refinement schemes in Relion, we were able to resolve the bases of these radial spoke structures. These showed that actually these two radial spokes per 96 nanometer repeat are molecularly distinct. They have a different subunit arrangement. It also revealed how these bind onto the doublet microtubule. So there's really long coiled coils that are 96 nanometers long that bind to the surface of the doublet microtubule, helping to establish the overall periodicity of the doublet microtubule. And then there are adapter proteins at the bases of the radial spokes that recognize specific sequences within this coiled coil to precisely position the radial spokes. And now we know the identity of these radial spokes, we can generate mutants to selectively remove one or the other. However, no matter how hard we tried, we couldn't resolve the kind of distal parts of the radial spokes, which were further from the microtubule surface. Well, we could, but we could only get about 5 angstrom resolution. So we needed an alternative approach. And for this, we decided just to strip the radial spokes from the doublet microtubules using a kind of high salt condition. We could then purify these individual radial spokes using anion exchange. We got a beautiful preparation and really beautiful 2D classes of this T-shaped complex. And then we used the whole arsenal of tools in Relion to be able to reconstruct this structure again to kind of mid 3-point-something angstrom. So this is what the structure of a kind of intact, mature radial spoke looks like. And again, using kind of very manual model building, we were able to build an atomic model of the radial spoke, identifying most of the components. And interestingly, when we compared it to the stalks, we found that we had selectively isolated only one of the radial spokes, radial spoke 1. And actually, if we look at how this radial spoke attaches to the doublet microtubule, it makes a lot of sense why we only isolated that one, because that binds via electrostatic interactions, whereas the other radial spoke 2, binds via a hydrophobic coiled coil. So of course, our high salt wash had only selectively removed radial spoke 1. We were also able to do some dynamic analysis using Cryo-Dragon and also rely on multi-body analysis, which showed that the radial spoke is very dynamic, can tilt on its stalk, and this probably explains why we couldn't resolve the spoke head initially on the doublet microtubule. And so we can put these models together to generate kind of a model of radial spokes attached to doublet microtubules. However, this was a little bit unsatisfactory because, of course, we're missing the axonemal dyneins. And if we really want to understand how this structure contributes to ciliary motility, we really need to have the axonemal dyneins as part of the structure, as part of the model. And so we had to go back to the drawing board and kind of come up with a different way of breaking the axoneme apart. And so we spent quite a few months developing a really gentle strategy to, instead of break the axoneme apart, just display it so that doublet microtubules over time kind of come apart from one another. And then these doublet microtubules are imageable by single particle cryo-EM. And using this approach and a combination of de novo model building and also AI-guided modeling, we now have a near complete atomic model of the 96 nanometer modular repeat. Now, this, of course, is a very complex structure. So we did what every English structural biologist would do, which was convert that into a London tube map, which really helps to illustrate the interconnectivity between these complexes. What we could also do was to propagate these doublet microtubule structures into this ring of doublet microtubules as they occur in the axoneme. But again, this was slightly unsatisfactory because it leaves a giant hole in the middle where the central apparatus, the central pair of microtubules should be. But we realized that as we were imaging the doublet microtubules, we also saw some central pair microtubules. So we went back to the micrographs and individually handpicked the C1 and C2 microtubules. And we were then able to use kind of processing to generate a structure and an atomic model of the central pair microtubules. And so we can now dock this into the middle of the ring of doublet microtubules to generate a near complete atomic model of the axoneme. This model contains over 5,500 copies of tubulin. It contains 165 different proteins, although now with Model Angelo, we're discovering a lot more that we didn't know before. So this gave us a lot of confidence to now go and tackle mammalian axonemes. But I was slightly concerned because we'd been growing clammy in liters in the cells. And so we decided to pick the kind of largest organ I could think of that was ciliated, which was the trachea from a cow. And the people in the lab were really excited to do this experiment, that they actually got caught by the police speeding back from the abattoir. Luckily, the police didn't investigate or search the car because they would have found a bunch of immigrants with some body parts, so which probably wouldn't have gone down well. But anyway, so once we had these tracheas, we could just brush off the cilia, demembranate them, isolate the axonemes, and then splay them apart into doublet microtubules. And this worked really well. And we were able to resolve the structure of the bovine doublet microtubule. And then we can compare that to the chlamydomonas structure. And this was great because it helped to identify proteins in common. And these are likely important kind of fundamental ciliary motility. But we could also identify kind of bovine and chlamydomonas specific proteins. And one of the biggest differences in bovine is that there's this bundle of tectin filaments inside the lumen of the A tubule. So we have a filament now inside a microtubule. And I find tectins a really fascinating protein. They're one of the most abundant proteins in cilia. They're also one of the most conserved. And they have this really simple structure where the second and fourth helix is exactly half the length of the first and third helix. And this is absolutely conserved through evolution. But until AlphaFold came along, no program could correctly fold this, which is really puzzling because it has a really simple fold where each helix just folds back on the other one. And this generates a kind of helical bundle with helical overhangs. These helical overhangs can then pair with neighboring molecules to form kind of long filaments. These long filaments can then pair with other tecton molecules, and they can form homodimers, heterodimers. They can have the same polarity, opposite polarity, incredibly versatile. And then these come together to form bundles. So it's a bit similar to what Bertrand was talking about yesterday with the threads and becoming ropes. And then we also identified a protein in the center of this bundle, which unfortunately we couldn't identify just based on the Cryo-EM maps. And so we had to really take advantage of the hyperstability of this filament. So we took our doublet microtubules, we added detergent, we added a denaturant, we kind of melted away the doublet microtubule, then we pelleted the surviving filaments. We did mass spec, and that helped to identify this kind of really unknown protein as being a tecton interacting protein. This really just demonstrates how we can go from cryo-EM structures to identifying kind of novel proteins. And so this approach with bovine doublet microtubules really helped to identify conserved factors and also bovine specific factors such as these tecton molecules. But again, I wasn't completely satisfied because really we want the structure of the human axoneme so that we can really understand dysfunction and how it leads to human genetic diseases. Now, we could have just used the same strategy that we used for the bovine and just waited for a human trachea to become available. I think people in my lab were a bit worried. I didn't have the patience for that to occur naturally. So we came up with this other strategy, which again, Victoria mentioned in her talk yesterday, where we take a nasal scrape brushing, which I think we're all far too familiar with. And then we use the cells isolated from that to grow two-dimensional organoids that kind of resemble the respiratory epithelium. And then from that, we can isolate the axoneme and then do cryo-EM on the doublet microtubules. And surprisingly, this works even better than brushing a dead cow part. And we now have a structure of the human 96 modular repeat of comparable quality to the chlamydomonas structure. So again, we can compare these structures. And if we just look at the doublet microtubules, there are differences between humans and cows, which really helps to kind of validate our desire to switch to a human model. But we also identify differences in most of the axonemal complexes. And in general, humans have simpler axonemal complexes than chlamydomonas. They have skinnier and radial spokes with fewer subunits, although there is a third radial spoke which may compensate for that. Their inner dynein arms are less diverse, fewer subunits. Their outer dynein arm is also simpler. There's really only one location where there's increased complexity in mammals, and that's the docking machinery that docks the axonemal complexes onto the doublet microtubule. We don't really understand the implication of this increased complexity, but when we looked closer, we saw that there were proteins, very small proteins, that linked this docking machinery with the internal MIPS. And this provided a great opportunity to test whether or not the interconnectivity between the microtubule was important for ciliary function. And so in collaboration, we deleted these two proteins in zebrafish and mouse. And in both cases, it led to kind of classic ciliopathy type phenotypes. This really shows that we can use cryo-EM to identify proteins that potentially could lead to human disease. And we think our structure, particularly of the human axoneme, is going to be very useful. It's going to provide a very useful reference for understanding the genetic causes of ciliopathies, such as primary ciliary dyskinesia. For example, our model contains 50% of all gene products that are known to be associated with this ciliopathy. And it will help to evaluate variants of unknown significance to speculate whether or not a mutation there could cause disease. But we don't only think that our atomic models can be useful for kind of evaluating known diseases, but we think that cryo-EM can be a useful clinical tool. And in proof of principle studies, we got access to 4 patients with PCD. These have known genetic mutations that cause kind of classic ciliopathy phenotypes. So the cilia are immotile or have reduced motility. The patients tend to be infertile and also have recurrent respiratory diseases. So we could take nasal brushings from each of these patients. We could then grow those cells into 2D organoids, isolate the cilia, and then do single particle cryo-EM on their structures. And without going into detail, these structures help to really correlate the genetic mutation with structural changes in the axoneme. And I think this could be a kind of blueprint for future personalized structural biology. And so just a quick summary. Hopefully I've shown you that we've now determined atomic models of the modular repeats of Chlamydomonas in human axonemes. This has helped to identify structural similarities and differences. We've shown that microtubule wall-spanning proteins that link the internal and external surfaces are important for motility, and their loss can cause ciliopathy-like phenotypes. We've established cryo-EM as a discovery tool for the de novo identification of proteins involved in ciliary motility, and we've demonstrated that cryo-EM can be used as an additional tool for diagnosis in ciliopathies, helping to provide insights into disease mechanisms. And I'd like to just end by thanking particularly my team in Travis Walton and Miao Gui, who did a lot of the work, but also my essential collaborators, particularly Ray Zhang Wastel, and also Hannah Mitchison and Tom Burgoyne in UCL. And thank you for listening.